Yair, but which is apt, as I hope you will see. So let me just get going here and establish whether this device of Ted's actually works. Look at that. You can hear me, you can see me, and you can see my pointer. I was, of course, not a student of Ted's, but an advisor of Ted's. I think I may be unique in this room in that position. But in fact, I was always a student of Ted's anyway. I had the great good fortune as an assistant professor to benefit from the fact that Ted had come to NYU after he left Michigan to work with George Sperling, with whom, as far as I can tell, he never shared a concordant thought. And after a while, I think he became a little tired of this, and he wandered up to my lab, which was on the immediately super adjacent floor, and asked if he could work with me. And I have to say that that was one of the best sort of happenstances that has come upon me in my professional life, and I owe a great deal to Ted, some of which I will tell you about today. So the theme that Yair gave me was related to this, I believe, terrible Broadway show, which I have never heard or seen, but which does actually exist. And I feel like I should point out at this stage something that Mike Morgan pointed out to me many years ago, which is that these things that I'm going to call plaids, and which the show also calls plaids, are not in fact plaids, but tartans, right? Plaids are multiplicative, tartans are additive, and actually that turns out to matter a lot for the analysis that we've done. Anyway, this was Ted when he was in my lab. This was how he arrived. This was how he was a few years later, when he abandoned me and NYU and went off to Sarnoff, I think, which you'll hear more about in another little while. For those, if there's anyone in the room who hasn't actually seen a moving plaid, this is how you build them. You take a grating and you add it to a second grating, and then you get a compound pattern whose motion is not the motion of either of the components. And this seemingly simple thing turns out to be extremely powerful as a tool for trying to understand how vision actually works, or how visual processing works when faced with the compound problem of looking for both the orientation and the motion content of objects in the world. So Ted framed this problem very nicely in terms of what's actually mistakenly called the aperture problem, observing that when you have two simple figures like these diamonds, one of which moves down, the other of which moves to the right, the motion that you measure on a local piece of the edge is in fact identical in the two cases. This is because the motion that you measure only is the orthogonal component of the motion, not the parallel component, the component parallel to the edge. You can solve this problem in several ways, including isolating special points and measuring their motion. But a simple way to do it is what Ted formulated as this intersection of constraints idea, where you measure the motion of one edge, you measure the motion of the other edge, and the motion which is compatible with both edge motions is in fact the motion of the object. And instead of using diamonds and things like that, since I came from Cambridge, and in Cambridge you don't use geometric figures, you use extended stripy patterns, the experiments that I'm going to talk about were all done with plaids, which were, generally speaking, composed of additive pairs of gratings, sometimes more than two, and you follow the same geometry of motion as this stuff. Now, Ted and I wrote in the end 3 papers based on the work that Ted did in my lab. There was a psychophysical paper which reported some observations about how the perception of these plaids worked, actually 2 psychophysical papers, one ACM paper and one Nature paper. And then this kind of monster thing at the bottom right, which was a paper that involved not only work with Ted, but also collaborations with Martin Gietze and Bill Newsom, including the neurophysiology, which I will spend the rest of my few minutes talking to you about. But I've been making a living off this stuff ever since. After Ted left, I published a few more papers on plaids, and then I actually published a few more papers on plaids. The most recent of these were published in 2019. So this is like a deep vein that I can continue to mine, as far as I can tell, for the rest of my career. We occasionally have become adventurous, also based on something Ted and I did, and measured 3 gratings together, a triplaid. I'm not going to spend any time talking about triplaids today, but there are some fun things you can do with those. But I'm a neurophysiologist, and my interest was not so much in the perceptual phenomena, but in the question of how these patterns and the way we perceived them reflected the underlying neurophysiology. And I actually still remember, I think it was an optical society meeting when Ted was in the lab, when either Ted or I gave a talk about these patterns, and Fergus Campbell, who was not given to enthusiasm about almost anything, came up to me afterwards and he said, I'm not going to do the Scottish accent well, but he said, "You've got a tiger by the tail." I was, all right, well, okay, tigers are stripes, two tigers make a plaid, whatever. But it's proved to be true. It's actually one of the few things that I think I can firmly attribute to Fergus, which I still believe, but never mind. Okay, so we were interested in how these plaids might influence the responses of directionally selective neurons. Horace Barlow had discovered directionally selective neurons in the retina, in the rabbit. In the 60s. In cortex, Hubel and Wiesel had described them a few years later. This is an example set of responses from one such cell, responding preferentially to movement in one direction, not the other. In visual cortex, primary cortex, about a quarter of the cells have this property. But in this area MT, which is an extrastriate area involved in motion processing, almost all cells have this property. And this is an example of a polar plot of a direction tuning curve of an MT cell. And virtually every cell in MT has a tuning curve that looks like that. Now, the question that we could pose, given the sort of plaid phenomenology, was a simple one, which was to ask whether the motion sensitivity of a cell was determined by the overall motion of the pattern as we perceive it in the plaid, or by the motion of its independent components. And so, since the pattern is additive, you can add the two gratings, the result is a plaid. But if you ask cells what they respond to, so if you take a cell that responds best to a vertical grating moving to the right, this would be its tuning curve, and you then ask how it responds to a set of plaids moving in different directions. If it responds according to the rules of the orientation components, then it will have a tuning curve with these two lobes, rabbit ears, if you will. And if it responds to the overall motion, it will be what we call a pattern direction selective cell, and will have a tuning curve that matches that for gratings. And so we went on to look at these properties in many areas in several different species. This is a typical response of a V1 cell, showing the two-lobe behavior. Here's the tuning curve for a grating, and for the plaid, there are two peaks of the tuning curve, one corresponding to each of the underlying gratings. Around this time, I presented some of this work in the hearing of Bill Newsom, then a postdoc at NIH. He said, well, whatever it is in cats does this. A right-thinking neuron in a monkey won't behave like this. And so, this was a challenge to him and to me. So he came to the lab and did experiments with us on the cells in MT, which is the area he was then studying in Bob Wertz's lab. This is an example of a component direction selective cell in MT. Again, a single lobe curve on one side, two lobes on the other. But he was correct in that MT contains a sizable fraction of cells that are pattern direction selective. That is to say, their tuning curves for gratings and for plaids are well matched. And you can measure this by comparing the accuracy with which you can predict the tuning of a cell based on the model that it's responding to the overall motion, which is the pattern prediction, or to the individual components, which is the component prediction. You can plot data from individual cells in this space, where you look at the correlation between the data and these two predictions. V1 cells are component direction selective. MT cells have a much wider dispersion and are often pattern direction selective. And in later experiments, Bill and I showed that the cells in V1 that project to MT also are component direction selective. They are specifically like other cells in V1. Now, at this stage, you might wonder why it is that this is interesting, because most people have the intuition, as Bill did when I first introduced it to him, that if a cell liked things moving to the right, it should like anything moving to the right. But that property of being invariant in direction tuning turns out not to be easy to achieve, and I'll show you why. These are the tuning curves of that one example cell I showed you, on the left for gratings and on the right for plaids. Looks perfectly natural. The dashed curve on the right is the tuning curve you would have expected for this cell if the plaid were presented and the two component gratings each evoked the same response that they had in isolation. So the first thing to note is that the optimal plaid over here, all right, is composed of two gratings which individually evoke very weak responses. So the best stimulus for the cell when you make a plaid out of them is a compound of two things that are both ineffective by themselves. What's worse is that if you take the best grating and you consider the two plaids to which it belongs, which is represented over here by these two vectors, the response of the single grating to the single grating is actually substantially higher than the response to either of the plaids containing that grating. So what you have to be doing is you have to have a compound set of operations which are enhancing responses to appropriate combinations and diminishing responses to inappropriate combinations. And we spent a while trying to think about how to model this. And modeling this turned out to be awkward. The intersection of constraints idea, which I mentioned earlier, which Ted had introduced to me, doesn't really have good stuff in it to make a neural model out of. It, for example, requires a logical AND operation, and neural circuits are not notably good at doing things like that. What we wanted was something like the model that Hubel and Wiesel had developed for understanding orientation selectivity in V1 cells, which you're probably all familiar with. And after an unduly long wait required to allow Arrow to pass through puberty, Arrow and David Heeger, in Arrow's thesis work in Ted's lab, came up with a model which I'm representing here as a set of green balls, which Arrow can explain later if you wish to understand them, but we will talk about it as the green ball model. It's a model of a linear feed-forward network that would respond best to motions of the kind that drive pattern direction selective cells well. And so this idea, Ted was not an author, I've always noticed on this paper, but presumably a favorable spectator. But Arrow and David came to me with this model in the mid-90s, and I said, well, that's very nice, but it won't actually work because you can't. And then they showed that it did sort of work for a whole bunch of data they pulled from the literature. But the task of actually fitting the model to data was one that turned out not to be easy, and which waited for the arrival on the scene of Nicole Rust, who was a student of mine and then a student and a postdoc with me at Arrow at NYU. And the model that she helped fit is basically this green ball model, but built into a more or less now standard framework of a V1 model, which has a sort of gain control stage in it and a linear input, and then an MT model, which also has a gain control stage and a linear input. And the idea, which many of us like, is that Cortex does the same thing over and over again, and that this model captures that simply by altering. I have a clock here, Yair, so you can't fool me, right? So these are the tuning curves of two more example cells, a component cell at the top, red is gratings, red is plaids and blue is gratings. Down here, a pattern direction selective cell. In order to test this model and to evaluate it fully, what we decided to do was use a larger stimulus set. And this is the stimulus set of all possible plaids. So these spaces here represent combinations of two gratings with all possible combinations being represented. The lines and marks on here represent special places, but I'm going to show them to you in animation rather than with words. The diagonal, which is where all gratings have, both gratings that make the plaid are the same, is simply a set of single gratings of double contrast. The red line here, this red diagonal, is a plaid with a 120-degree angle between the components, and its direction is changing as the bouncing dot runs around the place. The line which is here indicated, not drawn, but corresponds to a plus in this space, is the space of all plaids that contain one component that has a 180-degree orientation, that is to say, is a vertical grating moving to the right. So if you were a cell that liked vertical gratings, I lied, moving gratings to the left, vertical gratings moving to the left, then you should respond to all of these plaids. And finally, here along a negative diagonal is a set of plaids that have a common axis of motion. So it turns out that without getting your geometry engine too fired up, this is the whole space. This is what happens when it's late at night and you have something else you don't want to do. And so you end up making animations like this. This is about as many plaids as you can put on a single screen. In any case, these are data from two cells, a component direction selective cell and a pattern direction selective cell, that show how tightly they correspond to the predictions of this model. The component direction selective cell responds to all patterns that contain at least one component at the preferred orientation and direction at 180. And the pattern direction selective cell is extended along this negative diagonal axis, which is the axis of common motion. And this is work that Nicole Rust did. Now, this is actually the way we tested the model. The way we actually fit the model is way too tedious for a Saturday morning, but it was basically fitting what's called a linear nonlinear cascade model. But the tricky part of this model is that the input was not pixels, which is the way we're used to thinking about these models, but actually the inputs of an unknown set of direction selective cells from V1. To test that model, or to fit it, we used the ultimate plaid. This is a hyperplaid which contains no less than 6 components, each of which changes every 160 milliseconds. I'm not even going to try and justify that choice. But the bottom line is, if you look at the top, these are 5 cells from MT in this space. The response is plotted in this space. And at the bottom are, I'm going to say, parameter-free fits of the model to the data. Of course, that's always a lie. There's always a free parameter, but I'm not going to tell you what it is. And the bottom line on that is that the model actually does a nice job of accounting for the properties of these cells. I will, because I only have 1 minute and 15 seconds left, I will skip the details of why this should be so, and simply say that what this model and its fitting has turned out to reveal to us is something which I think has general importance and general interest for many kinds of transformations of information as they go from the striate to the extrastriate visual cortex, which is the results you get result both from local properties of cells in the input. So there are certain properties that V1 cells have to have for this model to work, and there are certain ways in which they are combined in the downstream area, in this case MT, which also have to be true for the model to work. Now, it turns out that the soundtrack recording of this, I think, tedious show is available on RCA Records, and what I can do with it is simply rather crudely paste in the faces of the players who mattered for this enterprise. Ted, of course, me, not really, Arrow, looking particularly degenerate, and Nicole Rust, who did a lot of the work in that fitting. And so at that point, I will stop and thank you for your time and take questions. Or not, as I'm out of time. But
time for quick questions.
I don't have any questions. I don't see why anybody else should. So.
Okay, so thank you, Tony. Let's move on to the next speaker.